Our world is teeming with life, much of it invisible to the naked eye. We share our planet with a vast array of microorganisms, including bacteria. Bacteria inhabit virtually every corner of the earth, from soil to air. Some bacteria are beneficial, others cause disease. Humans have developed antibiotics to combat bacterial infections. Overuse of antibiotics has led to antibiotic-resistant bacteria, or superbugs. These superbugs pose a significant threat to global health. Scientists are exploring alternative approaches to combat bacterial infections. One promising approach involves bacteriophages, viruses that kill bacteria. Phages are natural predators of bacteria and have therapeutic potential. Phage therapy is gaining interest as antibiotic resistance grows. This essay explores bacteriophages, their biology, and their potential in combating superbugs. Bacteriophages are viruses that infect and kill bacteria. They are ubiquitous in nature. These microscopic warriors are highly diverse in size, shape, and genetic makeup. All phages have genetic material encased within a protein coat called a capsid. The tail attaches to bacterial cells and injects the phage's genetic material. Phages are highly selective, making phage therapy a precise approach to treating infections. The life cycle of a bacteriophage is a remarkable example of biological warfare at the microscopic level. It begins with the phage attaching to the surface of a bacterial cell. The phage's tail fibers recognize specific receptors on the bacterial cell wall, ensuring that the phage only infects its intended target. Once attached, the phage uses its tail to penetrate the bacterial cell wall and inject its genetic material into the bacterium. Inside the bacterium, the phage's genetic material takes over the cell's machinery, forcing the bacterium to produce more phages. The phage's DNA or RNA hijacks the bacterium's ribosomes, the cellular machinery responsible for protein synthesis. Instead of producing proteins essential for its own survival, the bacterium is forced to churn out phage proteins. These proteins assemble into new phage particles, complete with heads, tails, and genetic material. The bacterium becomes a phage factory, mass-producing copies of the virus that infected it. Section 4. The rise of superbugs, a looming health crisis. The discovery of antibiotics revolutionized medicine, providing a powerful weapon against bacterial infections. However, the widespread use and misuse of antibiotics have driven the evolution of antibiotic resistance, leading to the emergence of superbugs. Superbugs are bacteria that have developed resistance to multiple antibiotics, making them difficult or even impossible to treat with existing drugs. The rise of superbugs poses a significant threat to global health. As bacteria become resistant to more and more antibiotics, we are approaching a post-antibiotic era, where common infections could once again become deadly. The World Health Organization has identified antibiotic resistance as one of the biggest threats to global health, food security, and development today. Without effective antibiotics, many medical procedures, such as organ transplants and chemotherapy, would become much riskier. The economic impact of antibiotic resistance is also substantial, with longer hospital stays, higher medical costs, and lost productivity. Section 5 of phage therapy, a beacon of hope in the fight against superbugs. In the face of the growing threat of antibiotic resistance, scientists are exploring alternative approaches to combat bacterial infections. One promising avenue of research is phage therapy, the use of bacteriophages to treat bacterial infections. Phages offer several advantages over traditional antibiotics. As mentioned earlier, phages are highly specific in their targets, meaning they are less likely to harm beneficial bacteria in the body. Phages can also evolve and adapt to bacterial resistance, unlike antibiotics, which have a static structure. Phage therapy is not a new concept. It was first explored in the early 20th century, but its development was overshadowed by the discovery of antibiotics. However, with the rise of antibiotic resistance, phage therapy is experiencing a resurgence of interest. Scientists are now investigating phages as potential treatments for a wide range of bacterial infections, including those caused by superbugs. Phage therapy has the potential to be a powerful tool in the fight against antibiotic resistance, offering a more targeted and sustainable approach to treating bacterial infections. Section 6. Triumphs and trials, real-world applications of phage therapy. While phage therapy is still considered experimental in many countries, there have been several promising real-world applications of this approach. One notable example is the case of Tom Patterson, an American scientist who contracted a life-threatening antibiotic-resistant bacterial infection while on a trip to Egypt in 2015. After conventional treatments failed, Patterson's wife, Stephanie Strathby, an infectious disease epidemiologist, turned to phage therapy as a last resort. Working with a team of researchers, they were able to identify and administer a cocktail of phages that successfully cleared Patterson's infection. Patterson's case garnered international attention and helped to renew interest in phage therapy. Since then, there have been several other reports of successful phage therapy treatments, including cases of patients with cystic fibrosis, urinary tract infections, and diabetic foot ulcers. Clinical trials are underway to further evaluate the safety and efficacy of phage therapy for various bacterial infections. Section 7. The future of phage therapy. Challenges and opportunities. While the potential of phage therapy is immense, several challenges need to be addressed before it can become a mainstream treatment option. One challenge is the regulatory landscape surrounding phage therapy. As phages are biological entities that can evolve and adapt, the traditional drug approval process, which is designed for static chemical compounds, may not be suitable. Regulatory agencies are working to develop appropriate frameworks for evaluating the safety and efficacy of phage therapy. Another challenge is the need for further research to optimize phage therapy protocols. Factors such as phage selection, dosage, and route of administration can all impact the effectiveness of treatment. Additionally, more research is needed to understand the long-term effects of phage therapy and the potential for bacteria to develop resistance to phages. Despite these challenges, the future of phage therapy is bright. With continued research and development, phage therapy has the potential to revolutionize the way we treat bacterial infections, offering a more targeted, sustainable, and effective approach to combating the growing threat of antibiotic resistance.